Hello, everyone. I'm Alex, a summer research intern at the Mayan Lab and an undergraduate student studying bioengineering. Today, I'd like to talk to you about the project I've worked on this summer, adding RNA-seq analysis capabilities to the Playbook workflow builder. To begin, I'd like to give you some background about the Common Fund Data Ecosystem NIH Common Fund Program, or CFDE. The CFDE is a relatively new NIH Common Fund program with the goals of optimizing the reuse of datasets and tools developed by Common Fund programs and finding ways to combine data produced by these programs for synergistic discoveries. The program combines Common Fund Data Coordinating Centers, or DCCs, to work together to achieve the CFDE goals. One of the CFDE partnership projects is called the Playbook Workflow Builder. The Playbook is a framework that gathers information from various DCCs. It is supported by DCC tools, APIs, and databases, forming a network of DCC microservices accessible through a user interface. Within this network, nodes represent semantic types like gene sets, while edges depict the transformations or visualizations performed on these semantic types by different functions. The Playbook offers an easy starting point for researchers without coding experience to perform a variety of analyses. Here is a visual representation of the Playbook. You can see that various DCC tools are used to produce this interactive workflow system where users can follow many paths to analyze or visualize their data. So the question that we asked was, how can we jumpstart usage of the Playbook by the biomedical research community and make the Playbook useful for as many use cases as possible? It's important to start by discussing BioGPs, which is a web platform developed by the Mayan Lab. BioGPs is a system that enables users to analyze their RNA-seq data. Users can upload their data and then automatically generate analysis reports in the form of Jupyter notebooks. Building on this concept, we wanted to upgrade the Playbook with the ability to analyze RNA-seq data similar to the way users of BioGPs perform such analyses. Since BioGPs is widely used, we hoped that by adding this functionality to the Playbook, we would be able to draw in many users. Here is a visualization of the overall workflow that was added to the Playbook. Users upload their RNA-seq data tables as gene count matrices. This data is visualized with a PCA plot and a library size plot. Further analysis can be performed by computing differential expression between two groups of samples and then performing gene set enrichment analysis. In order to create these individual components, the Playbook development environment was used. This interface, which is described in the Playbook user guide, was designed by Daniel Clark. So thank you to Daniel for all of the guidance. And in this case, you can see what it looked like to move through the workflow in order to generate a PCA plot in the development environment. To demonstrate how the new components added to the Playbook can be applied to a real use case, we selected an example from melanoma. Melanoma is a deadly skin cancer affecting thousands of patients in the US each year. It's important that we find drugs and targets to combat this, and the Playbook can be helpful in doing so. This Nature Communications paper developed data that compares 3 different melanoma cell lines to healthy human melanocytes. Here, the geo-expression data from the study was organized into a table with 3 SKMEL28 samples and 3 normal melanocyte samples. A metadata file that contains the labels of these samples was also created. Both files were uploaded to the Playbook so the cancer cells could be compared to the healthy cells. To display how executing this pipeline would look from a user point of view, here is a screenshot of the workflow in the Playbook app as the GeoStudy was analyzed. First, the input file card was selected and the data and metadata files were uploaded. From there, the files were merged into an AND-data file. As a user moves through the workflow, the analysis can be visualized on the top of their screen as shown here. If a user clicks on any icon representing a previous step of the analysis, they can move back to that step and create a new branch from there. As the user constructs the workflow, A description of the methods and the analysis is automatically generated and added into a paragraph with citations. This paragraph can be used when writing a paper based on the results obtained from running the final workflow. Then a PCA plot of the samples was generated. The user can click the PCA card, and then this is what the visual output of executing the code behind this card looks like. A user can also generate a visualization of the library sizes of their dataset by clicking the library size card. And then this is what the bar plots look like for this case study. The next step is to compute differential expression between the SKML28 cells and melanocytes. This can be achieved by either the characteristic direction or Lima-Vu methods. Here you can begin to see the branching capability of the playbook, where many routes of analysis can be created in one workflow. Next, the up or down regulated gene sets can be extracted and then visualized as a volcano plot using either differential expression method. Enrichment analysis was then performed on the differentially expressed genes using the enricher platform. From here, specific gene set libraries can be selected from enricher. And as a last step in this workflow, results from a selected library can be visualized directly in the playbook with the bar plot component. So now what did we find? This figure shows several bar plot results from enricher. One thing we observe is the upregulation of targets of the transcription factor MYC. MYC is a known oncogene that is important for regulating the cell cycle and is a critical transcription factor in development. Many of these results draw from common fund programs, including LINCS, HubMap, GTEx, IDG, and Glygen, and can be helpful in furthering melanoma research. In summary, we expanded the capabilities of the playbook to include RNA-seq analysis. The next step is to develop a component to label samples as control or perturbation, as shown on the left. We are also developing a Chrome extension that will add a button directly near listed geostudies to direct users to the playbook, as shown on the right. This will enable the reanalysis of published studies listed on the GeoDatabase web portal. And lastly, I'd like to thank everyone who was supportive of this project, especially Daniel, Dr. Mayan, and all lab members. Also, please leave any questions you may have in the Q&A feature. And I'd now like to hand it off to Lauren Malik as she describes how she created a new tool to perform enrichment analysis for long non-coding RNAs. Thanks, everyone.